We'll go right to the home page. All right, so on the Genome Browser homepage, we've got a couple of quick links that will work very easily. The main one you want to go to first is the one right here that says Genome Browser, although there are some things up here that look very tempting that say the same thing, Genome Browser. But these are actually quicker links to get into the browser. So I want to show you this one first. So if you go to this thing, it says Genome Browser, interactively visualize genome data, and click it, you get to a page that says Genome Browser Gateway. This is a nice place to start so that you know that this page exists, and also so that you can choose different species that are non-traditional model organisms, or not human or mouse. And you can get a little bit of information about the genome you're looking at yourself in terms of some specific ID numbers regarding the generation of that genome project, for example. And there are lots of different species for which genomes have been assembled, because we are in the age of high throughput genome sequencing. So if you really want to go look at the tarsier genome, or the genome of, I don't know, the southern sea otter, the Chinese pangolin, you can go look at those too, just like you can browse through the human or mouse genomes. Those genomes are going to be much less annotated. The assemblies are going to be less perfect because those are not huge model organisms. Those have mostly been sequenced for comparative purposes, for studying ideas about gene evolution. So these links, though, over here, genomes or genome browser, I'm going to just tap on them real quick to let you know that there's a pop-down menu where you can see the different species listed here. Human assemblies, the HG38 is the most recent human assembly, so that's the one you'll probably go to first. The HG19 is one that was released, I think, in 2009, and so it's not really used as much anymore. Here's the latest and greatest mouse genome assembly. The SARS genome is here too, and other will basically bring you back to this page where you can see all these other species here that you can pick from. And of course, the popular species are up here at the top. Okay, so there's a lot of different ways you can find a location in the genome. So here we've got the human genome December 2013 HG38 assembly selected. Where do we want to go? So you can type in a gene name. You can also type in coordinates if you have them handy. And in this case, there's a default gene that is already on the browser. So if you don't even type in anything and just hit the go button, you'll get some defaults that, so it'll bring you to some default gene. Usually it's some interesting gene that is people think is currently interesting. In this case, it's brought us to the ACE2 gene, right? And you guys may have heard of this gene. This is a gene that is a receptor for the coronavirus spike protein. So it's currently a gene of intense biological interest, right? Now, that's great. I want to go see a gene that I am more familiar with. So I'm going to type in a gene name here, BMP2. So this is a gene I've studied a lot. I want to go see where this gene is in the genome. So just bear with me for a second. It's going to look at its databases to see if it has anything that has the letters BMP2, and it has 3 genes that have that in the name, and BMP2 is the one we want, not these other unrelated genes. So we're going to click on that, and boom, it refreshes the page and brings us to the map of the BMP2 gene on the genome browser. Now, if you haven't used the browser before, you might get a little overwhelmed with all the information that's displayed by default, right? I mean, there's a lot going on here. We've got this thing that looks like a gene map that I've told you about already, but we've got gene expression, this thing called encode regulatory elements, histone marks, and this multi-Z alignment of 100 vertebrates. What's this? SNPs, repeat masker. So these are some tracks that people frequently are interested in looking at, but it's kind of like drinking from the fire hose, right? It's information overload. We can't talk about all of this stuff all at once. So let's just look at some basic controls on this browser to get oriented. And the very first thing I'll bring you to are these track buttons down here, which configure the whole display all at once. And this little button that says hide all, I'm going to click, which does exactly what it says. It turns off all of the tracks. All right, so now we're just looking at nothing. We're looking at that same position of the genome where BMP2 is, but no tracks are being displayed. In other words, we're just starting from scratch. And I kind of like doing this so we can just turn on the tracks that we're interested in and start from there. So I'm going to do that by bringing our GenCode data back. So how do we pick a track like GenCode? All of the tracks have individual control buttons. They're all grouped by categories, and GenCode annotation is in the genes and gene prediction tracks. Now, there's lots of different tracks because lots of people over the years have been interested in trying to annotate genes in the genome and predict where they are and where they begin and end and why. So there's lots of slightly different variations on this theme, but the GenCode one is, a widely accepted in sort of all-inclusive description of genes in the genome, and that's a great place to start. So I'm going to reactivate this track. If you tap on the pop-down menu, you've got options. Instead of hiding it, you can display it in a sort of a denser or more full format or something in between. I particularly like this format called pack, where it takes all the genes, all the elements of this track, and it basically displays them sort of intelligently on the browser to try to not spread them around too much, try to fit them on the browser window nicely. So I'm going to hit pack, and then I'm going to refresh the browser, hit any one of the refresh buttons, and it will do exactly what you expect. It's going to refresh it. All right. So we're back to showing the description of the BMP2 gene here. It's got 3 exons, and it's a not particularly big or complicated looking gene. So this is why I'm showing this one to you first, right? Now, I want to tweak the display on the browser because I think this font is too small, and I'm getting older, my eyes are terrible. I need to see this a little bit more clearly. So I'm going to go to this track button here called configure and click it. It's going to immediately bring me to a page where I can change some very basic things about the image. And so all I'm going to change here is the text size. So I'm going to pump it up a bit and make it a little bit larger. But of course, you can change the width of the window and turn on or off various annotations on the display itself that are sort of generic things. Like if you like having these little light blue vertical lines, you can turn that on and off, that sort of thing. Okay, so I'm going to make the text size bigger. And as advertised, things are a little bit bigger. Okay, a little bit more clear to see. Again, everything is drawn to scale. The span of the BMP2 gene is, according to this annotation, exactly 12,561 bases in this reference genome. Your actual size may vary if you've got extra bases or missing bases in your introns, but this is the annotated size. So it's got 3 exons. The thin and thick parts refer to non-coding and coding regions. It turns out the start codon is right at the beginning of this exon, so you can't even see where the coding region begins. But we'll look at that in a moment. A few more buttons to know about are the buttons up here. And again, these are exactly like the buttons that Jim Kent designed for his nematode Intronerator browser way back in the year 2000. But they're basically just allowing you to move left and right along the chromosome or zoom in and out as if you're hovering over the chromosome looking at a specific bit. I just think of it as like, I don't know, it's kind of like Google Street View for your chromosome. So, all right, let's just zoom out to see if there are any other genes nearby. I'm going to zoom out, oh, threefold. And it turns out we haven't found any other genes yet. For this particular gene, there's not a lot of other protein annotated genes that are close by. So the view isn't very cluttered. Now, we can move left and right. We'll do that a little bit later. Another key thing to do is to zoom in on a feature. So I mentioned the start codon was at the beginning of this exon here. So to zoom in on a little more, we can use the zoom in button. So if I zoom in tenfold, I'll be somewhere in the intron. I think my exon that I'm looking for is over to the left. So let me hit one of these sliding buttons, the move buttons, and see if that brings me a little bit closer. Now it's popping into view. I think I need to hit this button one more time. There it is. You can just make out there's a little bit of a untranslated region at the beginning of this exon. Okay, so I'm moving around a little bit, getting homing in on my target. There was a comment last time about these arrows. And again, these arrows simply indicate the direction of transcription of the gene. These are not individual features. They're just drawn on there to indicate the direction of transcription of this gene. And so you can ignore them. They're put in the introns just because that seemed like it was a convenient place to put them when the thing was designed. So let's zoom in on something. And another way to zoom in is to simply go up to the very top line here, the scale line, and click and drag with the mouse. It'll pop up a little window like this, a little shaded window. And when you let go, you may get a window that says, okay, you're doing a drag and select. Let me tell you about how that works. Don't show this again, and zoom in, and boom, it'll bring me zoomed into that region I had selected. If we go in even further to this position in the start of the exon, now we've zoomed in so far, I'm looking at only 60 bases, right? And this is the start codon of the BMP2 gene. It's only about 7 bases into the second exon. There's an ATG here. We can see the actual DNA sequence displayed because we've zoomed in enough. And we can see codons displayed for the gen code annotated gene as well, the amino acids encoded by those. Okay, so there you go. You can zoom in and out, inspect things at different levels according to the kind of information that can be displayed easily at those different levels of sort of magnification. So there's a few other things to quickly notice here. Sorry, just have to close that door, keep the noise quiet. We've zoomed in so much. Let's zoom back out to see our gene again. So I'm going to zoom out at least 100-fold. And here's most of our gene. How about 3-fold more? And then we'll just slide it on over a little bit. Oh, wrong way. Let's go this way. How about that? Okay, I've got the whole gene back in view. Now, all the items you display in the individual tracks are clickable entities. So that's a really useful feature of the browser. If you want more information about a gen code annotation, just click on it and it'll bring you to a page with information about that gene annotation. So every annotated gene has a unique ID number, and this page has also lots of other links to, for example, genomic sequence, the official messenger RNA and protein predictions from this sequence, and a whole bunch of other links too. So we'll get back to this page in a minute, but I just wanted to make the point that everything's clickable, right? So if you are curious about what that is, just try clicking on it and following it, and you'll probably get more information about the thing you wanted. So to get back to the browser, I can either click the back button or just click the genome browser link, brings you right back to the last place you were looking at on the browser. And it remembers all these things as cookies. So these things are going to be waiting for you when you come back to the browser, unless you specifically um, clear that information. Okay, let's bring up a couple more tracks of information so you can get a feeling for that. And I think another interesting track to bring up would be SNPs, single nucleotide variants. So I'm going to do that. If we scroll down, we see different groups of tracks of information, phenotype and literature. For example, this is a good place to look for mutations that might affect phenotypes in the gene of interest, messenger RNAs, expressed sequence tags, old school RNA information. You won't find a lot of easy links to RNA sequencing information, usually because that stuff is um, it's so dense that usually don't want to look at all of the RNA-seq data from a specific source. And by the way, then you would have to choose the tissue source too, the cell line. So it gets really complicated really fast. Regarding expression, there are some links to, say, the GTEx project that has relative levels of RNA expression for genes. Not really so much a genome annotation track, it's just a launching point to get expression information about a gene of interest. Encode regulation, I find really interesting. We're not going to talk about that a lot in the module this year. Comparisons to other genome sequences, also interesting. We'll get to that a little bit later in the module. And finally, variation. So this is a track that annotates positions that are polymorphic, positions that in the reference genome can vary in other human beings. One of the main databases that is used by geneticists today for this variation is dbSNP, SNP database. So we'll pull up those tracks, and the default tracks for dbSNP, let's see, let's set this to the full setting. Nah, we better set it to pack. I think that's safer. And now we'll refresh the page and see where are single nucleotide polymorphisms that are known to exist in and around the BMP2 gene. And so the browser says, yeah, okay, I've got lots of those. Enough of them that it didn't really want to display them any less dense than the format you're seeing right now. So we can zoom in on these a little bit and explore them a little bit more. So I'm going to do that right now with, I think, this exon over here, the third exon of the gene. So let me pull that open by, or zoom in on it rather, by clicking and dragging. And now we've zoomed into that so you can see a little bit more information. I periodically have to pause, open up my kitchen door so my dogs can move in and out of the house. So apologies for that. So SNPs, right? Short genetic variants from dbSNP release 153. So like other publicly used databases, this one gets periodically updated as people discover new sequence variants. So at this point in our history, we now have whole genome sequence data from hundreds of thousands of human beings. And so those have been sort of exhaustively combed through to look for variations as compared to this reference sequence. And so this is one of the databases that holds all those annotations. Now, each of us human beings has several million SNP variants as compared to the reference genome, single nucleotide variants. They're scattered across our genome. And actually, most of them are fairly common in human populations, meaning most of the single nucleotide variations that you have that are different from the reference genome also exist in other people in the world at fairly common frequencies, like more than 1% of humans have those same SNPs. You have a few tens of thousands of SNPs, though, that are really, really rare, that you only share with like one in tens of thousands of other people. So usually the default will show you common SNPs, which are ones that are generally at a frequency of about 1% or more in human populations. So these are some of them. And To display this a little bit more fully, I got to explore this track a little bit more. So what I'm going to do now is click on this link. You have noticed all of these look like hyperlinks. So if we click on a track link itself, we will get a page where we can change all sorts of information about this track and tweak it a little bit. And actually, um, the reason this was being displayed in the dense mode is because there's a sub track that needs to be set to the pack view. And it is this one here, which is the common SNPs. Basically, if something doesn't make sense with the track, just click on this track link to this page and you can read more about how to configure it. So I'm going to change this setting to pack. And I think if this is on pack as well, we should see things displayed a little bit more clearly. And here we go. So this is the position of SNPs that have a frequency of more than 1% in human chromosomes as sampled from thousands of human beings that were sequenced. And these are the ID numbers for these individual polymorphisms in this database and the polymorphisms themselves. There's even color coding. So if SNPs fall within exons, we have a color coding. Blue means they're in an untranslated region. But if they're in a coding region, right, a single nucleotide could either be a sense mutation, which does, or rather a non-synonymous or synonymous mutation. Synonymous meaning it does not change the amino acid. Non-synonymous or missense mutation, meaning it does change the amino acid. And not too surprisingly, if it does not change the amino acid, it's in green. And if it does potentially change the amino acid, it's in red. So it turns out, for example, with this SNP right here, if you change the common allele A to a T, which is also a fairly common allele, that changes the amino acid. Let's click on this and get a little bit more information about this SNP element. So here's again the page for this entity. So there's lots and lots of information on this page because these are just links to other types of databases and things that have observed this SNP in human sequences. But this particular one in the BMP2 gene can have apparently one of three alleles. And I can tell you right off the bat that both the A and the T allele are fairly common. But the G allele is so rare, I'm not even sure who reported that and put it in the database in the first place. And that doesn't really matter. So the frequency of the A allele in human chromosomes as sampled in the 1,000 Genomes Project, which sampled over 1,000 individuals from all over the world, so populations from all different continents. This allele, A, has a frequency of about 23% in human chromosomes. The T allele is basically almost all of the rest of the chromosomes, about 77%. So what this means is that all of us have a pretty decent chance of being heterozygous or homozygous for either allele. And I absolutely guarantee that a lot of people in this class today have um, any one of all three of those genotypes. So this is a common enough allele. I can also tell you that this probably cannot have a major impact on human phenotype or reproductive fitness, because there's no way these alleles would persist in the population at those frequencies. So I think this is a neutral variation with respect to selection. That's just looking at the allele frequency. Now, common variants are very powerful for doing association studies with phenotypes. So even though this doesn't seem to have a profound impact on natural selection, it's possible it does affect the risk of a certain disease. This might be simply because it's close to some other variants that affect the function of this gene. But the vast majority of SNPs in our genome are probably neutral. Geneticists are, a lot of what they do involves trying to do statistical tests on all of these variations to pick out the ones that actually do have statistically significant effects on disease. Okay, if you, again, lots of other links to find more information about individual SNPs or other entities on the browser. Let's go back to the genome browser now, where I've got all these SNPs displayed. And let's see, let's look at another track, repetitive sequences. So now that we've learned about these repetitive elements, let's let's look at those. So those are annotated too. And the way these have been annotated is basically way back 20 years ago now, when repetitive elements started to be carefully understood and categorized as the Genome Project was getting underway. A database was created out of human repetitive elements. And the idea is if you have a sequence of a repetitive element, you compare it back to the human genome to find the locations of all the positions that are similar to that element, you might as well annotate them so that we know where they are ahead of time. And so that the track that displays those repetitive elements way down at the bottom is this thing called repeat masker. So I'm going to set this to the full configuration. You can only use hide, dense, or full. Refresh the page. And there's not very many elements being shown here, but there is one right here, which it is listed as a simple repeat element. So what's a simple repeat element? Let's zoom in on it and take a quick look. So I'm going to carefully click and drag. And it says there are two here. Let's zoom in a little bit more so we can see things at the base pair level. And what is this? What is a simple element? Well, what I'm seeing here is a lot of T, A, T, A, T, A, 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 T, A, T. Okay, so a TA repeat is certainly a simple repeat element, right? So the simplest types of repetitive elements are these very short tandem repeats of often 1, 2, or 3 nucleotides. There are hundreds of thousands of these all over the genome. And the big issue with these is that it's a terrible place to try to design PCR parameters, for example, because they're not very specific. Let's zoom out some more and look at some more repetitive elements. I'm going to hit the 100x zoom out button. So it should bring us to over 3 kilobases of information, right sort of back to where we started. That's not very much. Let's go out tenfold more. Okay. Now you can see I'm running into some other problems. So zooming in and out may quickly display too much information, right? I don't want to see these SNPs displayed like this. This setting is clearly inappropriate to try to sift through all these SNPs. So even though we're seeing more repetitive elements, I want to get rid of our SNPs. So I'm going to turn that track off. Fine. So let's hide the DB SNP track, hit the refresh button, and now we can just focus on repeating elements. So at this level, we can see there are more of these repetitive elements. Now, I have referred to simple elements as being these really short tandem repeats. Those tend to be pretty short. So that's why these are all very narrow here. Now, sign and line elements are these retrotransposed elements, which are scattered across our genome in hundreds of thousands of copies. And you can already see that there are several of these short interspersed nuclear elements in different locations in the genome, in the BMP2 gene. We also have some line element fragments over here, LTRs, which are similar to viral sequences, and even some small pieces of ancient DNA transposons. You'll notice too that the intron and coding regions are relatively depleted in these elements. And in the intergenic regions, we have more of them. I'm going to just zoom over a little bit more to show you and give you an appreciation for the density of these elements and their size. Notice we've got some of these short elements that are stuck in the middle of some of the line elements, and multiple kilobases of DNA that are completely comprised of this repetitive material. Um, so this stuff is all over the place and is something you should be aware of if you're grabbing pieces of DNA sequence to do other types of analysis. So that's a good time to maybe go back to the BMP2 gene and um, try to get some DNA sequence. So let's extract some sequence for the BMP2 gene. Now, first of all, at any moment, you can grab the DNA that represents what you're looking at in the browser window. And all you have to do that is to go up here to this button that says View. And if you scroll down 2 stops to the DNA link, click on that, you'll get a very simple window that says Get the DNA for whatever position you're looking at right now. And you have some ways you can retrieve the sequence. And so the formatting in upper or lowercase is very simple to fix. If you want to simply mask all of the repetitive elements to say, oh, I don't know, the letter N, that might be useful because if you're then going to do a homology search and a blast search or something like that, almost all of those alignment tools they just ignore an N in the sequence. So it basically blinds it to the repetitive elements. I'm not going to do that just yet. I just want to show you that this works quickly and easily. So if you simply press that Get DNA button, boom, you instantly get that sequence back. This is completely unannotated, right? So it's just raw sequence from the reference genome. So we can't see the BMP2 gene in this. We can't see where the repetitive elements are. I just want you to know that you can grab this genomic sequence at any moment. Okay, I'll hit the back button once and then twice to get back to my browser window. Okay, so maybe a more useful way to get DNA information for our gene of interest is to click on that GenCode annotation for BMP2 again. So you've already seen this, but this is the page for the GenCode BMP2 annotation. And there's a couple of sequence links here that are really particularly useful. So you can get the genomic sequence for the BMP2 gene corresponding to the start and end of the transcription unit itself, or you can get links to the messenger RNA or protein sequence. So of course, those are going to be different in size and different in content. So here's the amino acid sequence of the BMP2 gene itself. That's just the protein, but if you wanted to quickly get that, there you go. How about the messenger RNA? It says may differ from genome. Well, I would expect it might because it shouldn't have introns, but more to the point, this may be annotated in a way that would reflect, for example, RNA editing of bases, which would be a post-transcriptional change. However, most genes are not subject to RNA editing. But just to be safe, if you want the messenger RNA, you might want to grab that. And again, this is completely unannotated, but that's the messenger RNA as predicted from the genomic sequence and with respect to any other known modifications. And finally, let's go to this genomic sequence tool. So this one, if you're really interested in having the full genomic information, but sort of carefully separated in ways that are useful, is really good. So let's click that because it's a special page which allows you to extract the sequence of the genomic DNA for your gene of interest, but also with respect to the exon-intron structure, if you want that to be broken apart. Let me show you what I mean. So if you look down here at these check marks and radio buttons, you have options to retrieve the sequence in different pieces. So what it's going to do here, if you've got these boxes clicked, is that it will give you the exons and the introns, but it'll break them apart separately in the text document that's returned to you. We're going to leave this radio button, one FASTA record per gene. If you don't know what FASTA format is, it is a very, very simple text format for the gene of interest. So if we simply click on that as it is, it gives you the genomic sequence starting from the very first transcribed base to the very last transcribed base. But the exons and introns are not separated here. So um, although that's kind of nice for the transcription unit, we don't, we might want to have this information. So what if you were, for example, oh, I don't know, trying to design PCR primers that would amplify an exon completely. So you wanted to have the intron, you want to have your primers positioned in the flanking introns around the exon. You need to know exactly where those are. So let's go back and use this radio button, which says one FASTA record per region, exon, intron, etc. And we'll hit that button now. And what you will see is there's this little header line now for each separate piece. And in this case, I wanted the pieces to be the exons and the introns. So here's the first exon of the gene, and now here's the first intron. I can tell it's an intron too, or at least confirm that it looks like an intron because the first two bases are GT and the last two bases are AG, which is typical of an intron. Here's the second exon. Here's that ATG codon right around here. That's the start translation point of the BMP2 protein. We've got a big second intron. And then finally, the last exon of the gene is here, and it's a bit longer because it's got a 3' UTR. Okay, so obviously, if you save this sequence by just copying and pasting it into a Word doc or whatever, you've now got the separate sequences for each exon and intron. Um, so those are particularly useful for other purposes of like designing primers and probes and things. If you if you want more promoter or upstream sequence or downstream sequence, you can customize that as well. So there's lots of different ways to customize the formatting so you get exactly what you want out of your sequence. Cool. So we've got just a few minutes left. So what I'm going to do is I'm going to do a sequence homology search that is built right into the genome browser itself. What I'm going to do is something called a BLAT search. The BLAT search tool is here, and it's right at the top. A BLAT sounds a little bit like BLAST, and in fact, its name is derived from a BLAST-like alignment tool. That's what it means. Although it's not exactly like BLAST, what it is a tool that will take any sequence you give it and compare it to the human genome reference sequence and show you any regions of significant alignment in the genome. So it's very, very fast because it only has to search through the human genome. It doesn't have to search through all of organismal sequence space. So we could do an experiment, like, for example, let's take the BMP2 messenger RNA and compare it to the human genome. So here it is. Here's the messenger RNA. Basically, this is just the 3 exons stitched together. Now I'm going to copy this right into my clipboard. So I just did a Command C to copy it. And I'm going to go back to our page here where all of these controls up at the top are still clickable, and the tools link has the BLAT tool. So I'm going to click on this and go to this tool. Here's the BLAT search page, and we're going to BLAT right back to the human genome. We're going to use BLAT as a verb. BLAT meaning a BLAST-like alignment tool. Now, it's not exactly like BLAST if you've ever used that. This tool is designed to be very, very fast. It's great for looking for regions of perfect identity. So for example, if somebody gives you a piece of sequence and says, hey, this is exon 13 of the ACE2 gene. Can you go just quickly find this position in the reference genome? This tool is perfect for that. If you're looking for cross-species homology or um, sequence homology between gene families within the genome, it's not the most sensitive tool for doing that. You really want to use something probably more like BLAST through the NCBI interface, because this is designed for speed, not sensitivity. Nevertheless, we may see some interesting things if we type in, again, the BMP2 messenger RNA, which I'm just copying and pasting here. And then I'm going to hit the submit button. Notice there's an I'm feeling lucky button, just like Google has for the top hit, but we're going to use submit. And we're going to then see what it thinks about that. So what we've got here is the BLAT search results page. And what you're seeing here is a table where every row on the table is a significant match that this algorithm has found in the genome, right? Now, again, this sacrifices sensitivity for speed, but a few things to note. First, if you're taking a human sequence and doing a BLAT search back to the genome, the very first thing it should find is itself, right? It's a real human sequence after all. So in this case, that's true. So you will notice that on chromosome 20, there is a 100% identical match that starts with the very first base of our query sequence and ends in the very last base of our query sequence. So that's the BMP2 gene itself. And if you click on the browser link, it will bring you to this location where it says, okay, here's the sequence you just gave me from the BLAT search, and here's where it matches onto the genome. And it happens to match the structure of the GenCode annotation exactly, which it should, because that's where we got it. Okay, that's not very interesting. What I'm going to show you next is that there are some other hits, right? Why is that? Well, there's a few different hits. You'll notice that for each of these other rows in the table, the identity is not 100%. If you look at the length of the match too, it's not going to be as long. And this thing called a score, which is roughly the number of identical bases, is much lower. Now, for each of these rows, there's also a details link. So when you compare the BMP2 messenger RNA to the actual genomic sequence in the genome, so this is actually the details of that alignment, and it's a 100% match, which isn't surprising. So this page, when you click on the details, shows you all of the bases that were basically perfectly matched between your blatted sequence, your query in the genome. In this case, all of them are, because it's perfect. So we're going to then scroll all the way down. Here's the matching in the genomics context, just showing you where the exons of the BMP2 gene are. But let's go back and look at the second row, and then I'm going to let you guys go because we're out of time. So the second row of the table has this match, which is not very long, and it's on chromosome 14. Let's look at the details here. The details of the alignment. Here's our query sequence, and none of them are shaded blue, so this is not where it was able to detect homology, but there's a tiny bit of sequence homology here, and you can see this one little segment in our messenger RNA is really pretty similar to this location on chromosome 14. Okay, without looking at anything else, we'll just go back to the browser, click that link for the second hit on this table, and we'll see it happens to be inside a gene called BMP4. So this is the most closely related family member to BMP2, and it is on the opposite strand, so its direction is the other way on chromosome 14. But our match here is in the middle of this terminal exon of BMP4. Remember, this is going from right to left as we're viewing it. And this turns out to be the most conserved region of this family of proteins. Now, there's a bunch of other BMP family genes in the genome, but they're less closely related evolutionarily. And so the BLAT algorithm was unable to detect those because it's just not sensitive enough. We would have to do searches in other ways to pick up that sort of evolutionary significance. That doesn't really matter that much. For today's purposes, I just wanted to demonstrate you can find other things in the genome this way. For example, here's a sign element down here. So using the tools I just showed you, again, this is the very last thing. If I click on this DNA real quick, I'm going to grab the DNA for this sign element, select, copy, do a quick BLAT search on this sign element. And now it matches to hundreds of places in the genome. So, in other words, repetitive elements really are repetitive. There's lots of homology to these in all sorts of places in the genome. Okay, back to the genome browser, and I'm gonna actually I'll stop sharing today. So that's a lot for one day to go through, um, but I think I have to stop there.